Good evening, everyone. I'm Connie Lee. I'm President and CEO of Angioma Alliance, and I want to welcome you tonight to an atorvastatin trial update, but also some updates on what's been happening in general in the field, as well as some question and answers time with Dr. Awad tonight. And I'll introduce him in a moment, but I just want to encourage everyone to use the Q&A in order to ask questions. You'll see it down at the bottom of your screen for most of you. You just click on that and you can type in a question at least until it's answered. I'm the only one who gets to see it. So you can, spelling mistakes are fine. So you can just ask the questions there rather than in the chat because we may not see them there. So without further ado, I would like to introduce Dr. Issam Awad, who is the John Harper Seeley Professor of Neurosurgery, and he is the Director of Neurovascular Surgery at the University of Chicago. He has served forever as the chair of the Angioma Alliance, now I've forgotten what we are, the Scientific Advisory Board, and he is also the medical director of the CCM Center of Excellence at the University of Chicago, and it is always a pleasure to have him. So Dr. Awad, I'd like to turn it over to you.
Thank you, Connie. It's nice to really have this opportunity to connect with the CCM community. This is really what it is about, and we'll use the ongoing research as an opportunity to update everybody. I want to also introduce Agnieszka Stadnik, who is with me tonight, and she manages our human subjects research operations. That's all the biomarker studies and all the trials and trial readiness projects. So many of you probably know her, as well as Christina Piedad, who is unable to be with us today, but she is on top of the game as always with this disease and with helping you. So I think at the very beginning, Connie suggested that I maybe take 3 or 4 research advances that happened during this past year, despite all the COVID crisis that we all have been going through. This did not stop research contributions. So I want to basically tell you about a few things that have hit the literature that are important. I cannot, of course, cover everything. There are a lot of papers out there, but I'll try to tell you what I think some important things. One paper in particular, I think, got a lot of attention from our community. It was a big effort because it was the one in which we collected microbiome specimen on human subjects. And as you know, there's been a lot of work in mice on the microbiome. And what this study included is many of you at many sites that helped donate specimen. And then as a result of that, we were able to control your stools with those that do not have cavernous angioma and also look at disease severity. And many of you also gave blood specimens at our institution. So we were able to combine all of those in a paper that was published in Nature Communications. So this is a very impactful paper. And the summary of that research was basically that we confirmed the idea of a permissive microbiome. So what do we mean by that? That sounds like a kind of strange scientific term. Permissive microbiome means that people who have the disease need to have different gut in order for their lesion to form, meaning the gut acts as a facilitator for lesion formation. So this is a big deal. We have long known that in mice, we have colonies of mice that we can give them all the genes of cavernous angioma, and those mice don't get cavernomas. And they don't get cavernomas because they have resistant microbiomes. And while other mice we raise in colonies that are a little bit more dirty and they interact with other mice and so forth. And those mice have microbiomes that predispose them to get cavernous angiomas. So what we showed in humans is that you are the same as the mice. So basically, the folks that get cavernous angiomas, whether it is a solitary lesion or a familial lesion, they share a microbiome that predisposes to that. Now, how do we know that it is the microbiome that predisposes, and it's not just having a lesion makes your microbiome different? The fact was that we found the same microbiome in people who have solitary lesions. So they don't even have the gene. They don't have the familial disease. And there is no way a tiny little solitary lesion can change your gut. So it had to be that the gut is really what causes it. And in fact, that thing was the same in people of all kinds of disease severity. So that again argued for a permissive microbiome rather than the lesions causing your gut to be different. So what does that mean? That is a start. It's a start as an opportunity for us to know that the gut matters and that we shouldn't mess with the gut because it could end up messing with our lesions. So that's the only thing that I can tell you right now. In the mice, of course, we all heard the story that when we use certain food preservatives that are designed to prolong shelf life of food, these also activate cavernous angiomas and cause bleeding. So it's probably better to eat healthy and organic rather than to have supplements. And in our practice, we have seen many patients, some of you maybe, who the first bleed in your life was after you took a prebiotic or a probiotic. We have made the case that you shouldn't really mess with your gut. Just avoid probiotics, prebiotics, avoid all of that until we know more about how to make the gut healthier for CCM. And for now, the only thing we know is eating organic food rather than supplements. So that's kind of where we are. I know that's not enough. All of you want to know more. And we are doing a lot more work on this, and we promise you that if we have anything that helps us adapt to this, we will let you know. One bright light there was that the blood samples that you guys gave often reflect this permissive microbiome through certain binding proteins that bind to the bacterial product in our blood. So it may be possible that a blood test can tell you whether your gut is favored or not. And wouldn't that be nice, so you don't have to give stool specimen to know that? And it's a lot cheaper because, you know, these stool specimens are analyzed in a very complicated way with bacterial genetics. But if we have a simple blood test, it would be good. So the second broad area that I think was important this past year were several groups that reported new animal models of CCM. We, until now, had only one chronic model that was very complicated to produce. It was heterozygous, meaning like the familial patients, but we had to sensitize the mice in order for them to get lesions because the mice have small brains and they don't live very long, so they don't have enough time to form lesions on their own. That model is complicated because it takes about 8 mice in order to produce one that has a lesion. So it is a very inefficient type of model. And instead now we have ways of inducing lesions using the homozygous state. So it's not something we want any of you to have, being homozygous, but we can induce homozygous state after birth. So if you induce it before birth, the mouse dies. But if you induce it after birth, then every cell that divides could form a cavernoma. So these mice form a lot of cavernomas, and each mouse forms cavernomas. So, and they form them in the brain and spinal cord. So we're able now to test drugs much, much more efficiently. We can run 6 simultaneous drug studies with the same number of mice that we used to do before. So I think this is a big change. Several groups in Europe confirmed that, and they're already starting to use the same model. The other thing that that was reported this past year was also a very interesting fish model. In the past, the fish cavernous angioma model depended on heart malformations, which is not really something that matters to you, and it may not be applicable to the human disease. However, now the group in San Diego is able to produce actual cavernous angiomas in the fish. So we're not looking at the fish for heart malformations, but we're actually looking at multi-cavernous lesions. And some drug studies are now being done as a screening study. And then the drugs that are winning then can be taken to the mice, and then the ones that win in the mice get taken to humans. So that's kind of the thing there. The last thing I want to tell you is 3 big projects were funded by the NIH this past year. You may have heard that the BVMC study, which is the familial vascular malformation study, it includes familial AVMs, familial cavernous angiomas, and another disease called Sturge-Weber disease. But for us, for our disease in cavernous angiomas, this basically studies the families. And this was funded for another 5 years. And I think in this next 5 years, it is going to make a lot of progress on issues like blood tests that can help us decide which patients will form lesions, and also other biomarkers and factors in familial disease in general, and comparison of genotypes, because the first 5 years of the study were largely New Mexico based with common Hispanic mutation, but now we're going to bring in a lot of the CCM3 and CCM2 patients into it. The second big study that was renewed for another 5 years, and it's a huge undertaking at the NIH, is the program project that is run by Duke University, UPenn, UC San Diego, and University of Chicago. So this is basically the leading biology program for cavernous angiomas in North America. And it is the one that produced the microbiome stuff, that produced the bleeding in the brain analyses, and a lot of things came up in the last 5 years as a result of this effort. Now what we are doing in that project is for the next 5 years, going over human lesions, ones that I resect in Chicago primarily, because immediately upon resecting the lesions, we're going to take cells from that lesion and do very advanced molecular biology on the cells. The idea being single cells from the lesions can tell us a lot of story that the whole lesion combined couldn't tell. We're also going to take a plasma specimen from the mice. We used to throw away the mice except for their brains, and now we're going to take their plasma, and as a result of that, we're going to use the mice as a way to help us develop the blood tests that we will take to humans. The third project, and this is the last thing I want to tell you about in terms of novel research, is a very large grant that we received to work with Mayo Clinic and with UC San Francisco on blood biomarkers of symptomatic hemorrhage. Why is that important? everyone that sees me asks, did I bleed last year? Am I going to bleed next year? So we have a blood test that's very close to being able to answer that question. But until now, that blood test had only been done on Chicago patients. So now we will be able to confirm the results and even optimize the blood test with additional new molecules in collaboration with cases enrolled in San Francisco and Mayo. And as a result of that, we will be FDA able or FDA certifiable test that we hope a company will be formed to produce it as a blood test, or that the blood test will be purchased by a lab company to develop it for routine clinical use. But this is a huge deal, and it's another approximately $4 million investment by the NIH to help with blood test of hemorrhage in the brain. And I want to also tell you that we are following the signals that tell us that these blood tests in cavernous angioma may actually be useful for older people who do not have cavernous angioma, because as the brain ages, the blood vessels start to weaken in very similar ways as cavernous angiomas. So that is another very exciting venue. So thank you for letting me kind of give you a little broad horizon update on what I thought were exciting things. I'm sure there are a lot of other things going on, but these are them. I think the program at the investigator workshop that's being held virtually this year is extremely exciting. Many of you have not seen the program yet, or soon will, but it really is amazing. There are a lot of advances on these and other topics, including some things that I cannot tell you about because they will be presented at the meeting, but they are really very exciting results. As far as the atorvastatin trial, we have had several very important milestones. So we completed only about 3 months behind schedule the enrollment of our 40th patient. So this is really amazing considering the COVID crisis. As you know, we are aiming to enroll 80 patients, and we had hoped that we would enroll the 40th patient in the beginning of 2020. It ended up happening closer to late spring. And we are now at what? Are we 45, Agnieszka?
46.
46. So we are moving ahead. We continue to enroll at a very, very good pace. We just enrolled a patient last week, one the week before. And the idea here is to really complete this trial. This trial is very important, not because you all need to know whether statin is good or bad for you, which is very important to know, but also to show the world and the pharmaceutical companies that we as a community can do a trial in this disease. If we are resistant to even try something like statin, how the heck are they gonna hope that we're gonna try a drug that's never been used in human beings before, you know? So it becomes a challenge for us not to just talk the talk, but to walk the walk. We've been begging for drugs, and the way drugs are shown to be effective or not is by being tested, not just by being an idea in the mice. So this is my push for you guys to, if you have a cavernous angioma with symptomatic hemorrhage, please contact the CASH program. And we will make every effort to accommodate you with all the safety measures that we are employing at the University of Chicago for the COVID issues. Chicago is better than most of the country right now. University of Chicago is doing extremely well. We have not had any COVID acquisitions among patients during this pandemic, which is an amazing feat, and we've been watching this very, very closely. That means that we are doing all the safety measures, and we are very comfortable with working with you and with everyone else to keep treating the disease and not let the disease fall behind. I want to make a particular appeal for the follow-ups. So the biomarkers are how we will get the answer in this trial. There are not enough cases in the trial to count bleeds. So far we have had about 3 or 4 bleeds. Actually, there is no way that can be different between statin and non-statin. However, there have been very exciting changes in the biomarkers. The biomarkers have performed at better than 95% success rate, which we had projected that they would perform at 75 or 80%. That means almost every MRI that's done in our hands is being usable to answer the question. That is a very big feat. And it may help us answer the question with fewer than 80 patients because of the efficiency of the biomarkers. We just completed a big statistical huddle this afternoon with our statisticians, and we are very encouraged that there may be ways at the midpoint of the trial where we do some analyses and are able to project that the trial can give us an answer with maybe less patients than the 80. But that will never happen if we don't have 1-year follow-up and 2-year follow-up scans. That means for now, those of you who signed up, take the $500 and come overnight to Chicago. That's all I can tell you. We pay you to come. Unless the MRI is being done for clinical reasons too, the MRI is free. So if you don't want to have a clinical visit, we'll just do a quick research visit. You walk in and out. And that's, it's a shame for you to be signed up to a trial and not to do the biomarker that will help us get the answer. It's just, it's something that is not doable. There are several of you that say you are uncomfortable with traveling, and we respect that, but we are all trying to be heroes to help for this disease. And I can assure you, you are not taking unnecessary risks, because we are very much aware that the benefit you will get from the answer far exceeds the risk of catching COVID on our watch. So that's what I wanted to say. There is one thing that Agnieszka wants to share with you, that we have managed to invest some funds to develop something that will be very neat for this and future trials. So Agnieszka, want to tell them a little bit about this, and then I will put a final word on the trial readiness initiative, and then we'll go to the Q&A. Go ahead, Agnieszka.
Thank you. Hi, everyone. So all of you who are on the trial currently know that you have to log every day you've taken your medication and send us back the calendars and the bottles, and we call you every 3 months, and then there's further follow-ups if you have symptoms or anything. So we're actually developing an app. We're, I hope, less than 2 months out from actually implementing it, which would allow you to, first of all, remind you to take your medication every day, so there'd be no missed doses, hopefully. Also, it will allow you to enter any symptoms if you have them, or questions, to contact Christina or I directly. And it'll also replace the quarterly calls with Christina. You just do it over the app. So assuming you have a smartphone, which I think everyone does at this point, but if not, we can work around that, obviously. So it'll be just a better way to...
Whoever doesn't have a smartphone, I'll buy them one.
All right. Everyone's recording, everyone heard it. So yeah, it'll just be an easier way to interface with us, a little more with the times, instead of doing the paper and phone calls. So like I said, we're hoping within the next 2 months we'll have that available. And once it's close to up and running, we'll be in touch with everyone to get you switched over to the app versus the paper and follow up that way. So we're pretty excited about it. Hopefully it'll be beneficial to everyone who's in the trial or thinking about joining the trial. It'll be a little less work for everyone too. Yeah.
Thank you, Agnieszka. So there is another thing that's kind of exciting, but also a mixed news because of the COVID situation, which has to do with the trial readiness project. So the trial readiness project involves people who have CASH, meaning symptomatic hemorrhage, and who are seeing one of the doctors at the major clinical centers or centers of excellence. And you undergo the same types of studies that you would as if you were in a trial, so that we have a very good way of designing the next trial at multiple sites. So the idea here is that if you're a Mayo Clinic patient or if you live in California, you wouldn't have to come to Chicago to be in a trial. The idea is that you could do the trial at any of the cavernous angioma centers of excellence or even clinical centers. So we've made a lot of progress in validating these MRI biomarkers at the multiple centers. And that has been really a superb piece of work by the engineers at all sites. So that now if you get a permeability biomarker, for example, scan in New Mexico or in Mayo Clinic or at San Francisco, it will be the exact same as you would get in Chicago. The problem has been that many sites have really not reactivated their research as much as they would want because of COVID. So except for Mayo Clinic and University of Chicago, most of the other sites are in COVID hibernation. So they've enrolled maybe 1 patient, 2 patients in the whole year. Now we've made up for it. And in fact, between Kelly Fleming and our site, we are meeting all the milestones of the trial readiness project. But it's sad that we cannot have Johns Hopkins and San Francisco and New Mexico and Utah also contribute patients. We hope that as soon as they are able to get back online, that the next drug after atorvastatin would not be tested only in one site. It would be tested at multiple sites, and we have all the evidence that even these complicated biomarkers that we all had a lot of doubts about. I know Connie and I had a lot of discussion whether QSM could ever be used. Now we showed that 98% of the QSM scans at multiple sites are usable, and the cost is very, very reasonable. So I think this is good news for the future. But again, it calls for people to kind of engage a little bit more in the process. So Connie, is there anything that I forgot that I should have mentioned among highlights that you and I have talked about, or anything else?
Yeah, I think you've really covered the primary ones that have happened in the last year. You know, that we have medications that are in development specifically for CCM, but they aren't really at a point where we can talk a lot about them. So in terms of the research that's been done over the last year, I think you absolutely covered it. Do you want me to go ahead and start asking you some questions?
Sure, absolutely.
All right, I'm going to start with the atorvastatin trial questions, since that's the primary topic of the webinar tonight. Someone asks, would off-label usage of atorvastatin be recommended for patients at this time? And are there any risks?
Yes, there are risks, and it is not recommended. So the reason for that, and actually this was discussed thoroughly by everyone who is aware of the animal data and who helped write the guidelines. And the reason for this is that we do not know really if atorvastatin can make a lesion bleed more. So we do not want people to take atorvastatin off-label for CCM bleeds or for CCM. We don't think that will be the case, but that's not enough unless we are able to show that it actually decreases bleeding. You may be doing more harm than good and not know. So why would we even tell you to be in a trial if there is a chance it could be harmful? The reason ethically is because there is a chance you would be on the other option that's better. No matter which one turns out better, you're now engaged and getting a 50% shot at that. However, if you were to take it on your own or not take it on your own, you'll be a loser if you made the wrong decision. So, and nobody knows. So if you think you can outsmart it, I can talk you out of it. You know, you can go to you do whatever you want. But the fact of the matter is we do not know. And the soonest we know, we tell everyone else to do it. So that's the reason not to take it off label.
Okay, thank you. All right, the next question is, if atorvastatin is shown to be effective, would any other statin also be effective? In other words, could you substitute, Crestor or one of the other statins for atorvastatin?
So the reason we are using Lipitor 80 in the trial, or atorvastatin 80 in the trial, is because at that dose, Rho kinase, which is the biologic signal that is active in cavernous angiomas, is inhibited in humans. So if you take simvastatin at the highest allowable dose by the FDA, which is 40 milligrams, you cannot reach ROCK inhibition. Only 5% of the patients have ROCK inhibition at those doses. Atorvastatin 40 milligram is still as good as Simva 80, which is not allowed. And therefore, if you cannot tolerate 80, the trial will allow us to take you to Atorva 40 just for tolerance. And some patients will have benefit from that. But it has to be a high dose statin. There is no other way to take a low dose statin and hope that it will have the benefit we're after. Now, there are stronger statins like rosuvastatin that's actually stronger than Lipitor 80, and it has more side effects than Lipitor 80. So I don't know why you would want to take it. I think people use those very powerful statins in extremely resistant atherosclerosis, where they have multiple stents that are progressing despite Lipitor 80. So they get put on these very high dose statin, but we have no reason to pursue that in CCM. Now, will there be a specific drug that does what statin aims to do? There are several drug companies working on that, at least two very serious about developing a ROCK inhibitor for CCM application, but it will be a drug that's never been used in a human being before. So it could make you grow horns, lose your hair, we don't know. Or it could be the miracle drug that melts everything away. But the fact of the matter is that if we are too shy to take Lipitor 80 in the first trial, I don't know how many of you are going to step up and take a drug that's never been used before. So that's the reality.
So, and I think you answered the question about what about alternatives for people who can't tolerate statins at all. It seems like it's wait until
at this point, yeah. So let me tell you, we have enough experience now in the trial about tolerance. We, half the people are taking statin, half the people are taking placebo. But so far, in the atorvastatin trial and in patients in trial readiness whom we know are on statin, there really have not been any serious red flags. We have very careful safety monitors that are looking at all this data. And in fact, you are more closely watched taking a statin on this trial than taking it on your own, because everybody's watching and making sure that you're gonna be okay. So some people have stopped the drug because of feeling fogginess or some achiness, or they just don't wanna be on a drug. And I don't know if they were even taking the drug or a sugar pill. In fact, in the big statin trials, those side effects are about as equal in the people taking placebo as those taking statins. So I just don't know. We just have to see how this plays out. But no serious side effects have been flagged.
Okay. All right, and then someone is asking about the purpose of atorvastatin. They wanted to know whether the intent of the treatment is to prevent a bleed. And then if so, the follow-on is that only for people who've had recent bleeds, or could it be for people who had bleeds in the distant past, or maybe never had a bleed at all yet?
Right. So the reason we're testing it in people who have bleeds is because they are the most likely ones to rebleed. Therefore, we are the most likely to see an effect of a drug. If we gave the drug across the board to people, many of them have no symptoms, it will be very hard to show the effect of the drug. Because if you're doing fine without it, taking it, you're only going to have side effects. You're not going to have benefit because you're not having problems. So this is why we're singling out initially the people with the highest risk to give the drug. There could be an argument to take all symptomatic people and see if a drug will help them, and there are some people pursuing that. But to give a drug to asymptomatic patients right now, we have to really show that it works in the high-risk group before you are able to say that I'm doing some good taking it. So I think if you have a cavernoma that's never done anything, or maybe it's causing seizures, but they're well controlled, there is really no way I can make an argument for you to take statin now. And certainly we're not studying it in that group.
And a very basic question, is atorvastatin expected to be useful for both familial and sporadic?
Yes, so that's very important and very interesting. And in fact, we fault the group in Italy for only studying familial cases they're studying propranolol, but they're only enrolling familial cases. That's because their basic scientists have only done mice on familial disease, you know. The fact of the matter is that the Duke group and us have shown that sporadic lesions, meaning the ones that are not familial, have within them the same gene abnormalities as the familial disease. It's just you're not inheriting it in your bloodstream, it only develops in that one spot by chance. But the lesion itself is biologically indistinguishable. Therefore, a drug target that affects the familial disease should affect the lesion that is sporadic. The only difference is that if you're taking a drug to prevent lesions from forming, that drug is only going to work in familial cases because people with one lesion don't form another lesion by definition. So that's a different question. So in CCM3, for example, we might be after gene therapies to restore gene function so you never get lesions. But we can't give that to the whole world so they don't get a single lesion, you know. So lesion prevention is only true in familial cases, but atorvastatin and most of the other drugs we're targeting hemorrhage will be useful in both lesions.
And someone else asks you to please explain a little bit more what a biomarker is. You use the term a lot, but I'm not sure that you unpacked it enough for us.
Very good. Before I answer that question, I want to also say that the gut results also indicate that any changes in the gut that activate cavernous angioma probably act in both familial and sporadic lesions. So it's very important that this isn't just in the familial cases, but it could have the same effect. So let's say you have an inflammatory bowel disease that's causing activation of cavernous angioma. It could possibly act on either lesion type. The biomarker is something that we measure in the clinic that tells us about the disease. So an example of a biomarker is hemoglobin A1C in the diabetic, or cholesterol level in people who have atherosclerosis. That means If that is high in your blood, your disease is active. If that is low in your blood, your disease is well treated. And we are after something like that in cavernous angioma. So we want disease severity tests. We want predictive tests. So we want, you know, if you had hemoglobin A1C that's 11, people are going to tell you you're probably going to shut your kidney and have a heart attack in the next 5 years. So we want to be able to have a blood test that tells you, person A, your cavernous angiomas are chilled. Go ahead, live your life. Or your cavernous angioma, whatever you're doing, there is something not good here. And therefore, let's start digging more into stabilizing your lesions. And I think the big drugs, pharma companies, want to have a test like that because they would love to take the people who are going to have a problem and give them their experimental drugs. So, and you would want that. I mean, so if you knew you were going to bleed next year, you'd sign up for anything. And I think that's kind of what we're talking about.
Thanks. And then folks are wondering how long the trial is going to take from now on, from this point out before you.
Yeah, so we are halfway in a 5-year trial. So that factors in potential delays in enrollment. So that will not delay it beyond 5 years because we factored in a very conservative estimate. So we're able to enroll patients toward the end of the trial to still meet the outlook. The likelihood of it finishing before 5 years is nearly zero because of COVID. So if everybody was as excited about the trial as when it started, we'd finish it a year early. But the reality is that we are enrolling on target, not above target. It's good that we're not behind despite COVID. So that's great, but it's a 5-year trial. It's funded for 5 years, and I cannot promise you results before then. There is another scenario whereby if we find a safety signal, the trial will stop, meaning you're bleeding more or there is a problem with taking statin, therefore we should stop the trial. If that happens, you'll have your answer sooner. There is also a remote possibility that at the midway point, we find out that we could maybe get the answer with following people for 1 year without having to follow people for 2 years. So that could shorten the trial by a few months.
Someone wants to know if they're already taking atorvastatin for treatment of high cholesterol, should they stop until the trial is done?
No. So the benefit of atorvastatin for cardiovascular disease and stroke prevention and MI prevention is already demonstrated. Therefore, you'd be harming yourself by stopping it so you get into a trial and then having a heart attack. So we don't want that. So the guidelines are very clear. Go to angioma.org CCM guidelines. And you will see that we say that if you have a cardiovascular indication for statin, you would not be in the trial, you'd be on statin. But we'll never be able to know if it's helping your cavernous angioma, but we want to prevent you from having a heart attack or stroke.
Yeah. Okay, and then the next question is, looking out into the future, once this trial is done, the EPIC trial is done, what's the next step after that?
Yeah, so the EPIC could be the end of it for statin in 2 scenarios. If it shows a safety problem, there will never be another statin trial. So in that way, we would have done a great service to the community by telling you that statin is not good for you. So then it would be the answer, the final answer. There'll never be more studies. If the benefit is compelling, it is arguable with a drug as common as statin, whether we can stop people from taking it. The right answer by the FDA would be that you have to do a phase 3 study in order to show a label effect, meaning it says that atorvastatin helps cavernous angiomas. It's written on the bottle. But you know, most people will probably take statin. I may prescribe statin if the results are so strongly favorable. But right now, we don't even know it's safe.
Mhm.
So that's why we're not on the bandwagon to prescribe it.
Um,
Oh,
so
other drugs. That was the other half of your question. I think. Would there
no,
be
I didn't
something else?
actually, I didn't ask
Oh,
that
all
one.
right. Okay. I thought you
Got
said
the preview.
what's beyond this trial.
No, no, but my question is, if atorvastatin turns out to be not successful, or even if it does, what are the next medicines that you would want to see tested?
Right. So if atorvastatin is not helpful, it's out. So that's good. If atorvastatin is helpful, I think it's going to create a little bit of a challenge for the pharmaceutical companies because they may have to test their drug against statin. And many do not want to do that because they want to just test it against placebo, and the FDA will allow them that. So there will be some drug companies that are testing a drug for cavernous angioma. I'll tell you which drugs in a minute. But testing a drug for cavernous angioma, regardless of potential benefit shown with statin. And then they will let the market decides and science decide which was stronger and which one patients want to take. This is not very different from rheumatoid arthritis, where you have very expensive drugs given IV every month. But also there are drugs like Plaquenil and hydroxychloroquine that are given for rheumatoid arthritis. So some people choose to take that drug, some people choose, and there have never been trials head-to-head among effective medications. The same with Coumadin and other blood thinners. There have not always been head-to-head trials comparing them. So people just decide to go for Eliquis. It's sexy, and they stop Coumadin, you know? So that's okay, and that's all right, and you'll have options. So I'm happy to have two drugs that work. You know, as a scientist, I'd want to know which is better, but as a clinician, I'm happy to have two that work. What other drugs? That's going to be a big question. So you all know that there is a trial going on in Italy. It had, unfortunately, a little setback because Italy was hit very hard with COVID, but they're back on. And it is a trial of propranolol, but it is actually a weaker trial than the one we're doing because it's not powered to show an effect. So it's only going to show maybe a signal, but not an effect. What that means is that you cannot show that the two groups are really different. There are not enough cases to do that. And they are doing it only in familial cases. And they're using propranolol at a dose that's probably lower than the one that should work for angiomas, if it works for angiomas. So that's going to be an issue. And I think propranolol may have a role to play, but it will have to be at a high dose that many people do not tolerate. And that trial in Italy is not going to answer it at the dose that it will help cavernous angiomas. So propranolol is there, but if people get dizzy when they take it and they don't want it at the dose that it works, then you have to look for some other analogs that are better tolerated. There are exciting drugs. There is Tempol, of course, which is a drug that is an anti-inflammatory drug, initially developed for baldness, but now it's being developed for an oral preparation specifically to help CCM. We don't know how well tolerated it will be in CCM patients. We don't know if it has side effects or not. The phase 1 studies are very limited. They're basically making sure you don't drop dead when you take it. But we're going to have to really look at a lot of patients taking it before we know whether it works and also the side effects. There are other kind of big guns, the anti-angiogenesis drugs like Avastin, drugs like rapamycin, and others that you will be hearing about are potentially very useful drug in the animals. But man, we're going to have to decide who is willing to step up and take one of those very big drugs that are basically cancer chemotherapy type drugs. And maybe somebody has a lesion that keeps bleeding and they get on it. But, you know, my experience in offering things is that you still have surgery, you know, and those people are not going to go take a chemotherapy drug if the lesion can be taken out, you know. So I think these are going to be issues, but there will be drugs. Now, if you develop drugs that are analogs that are better tolerated, that will be very good. And I think the market is very promising because the pharma companies now know that we can do trials at multiple sites. So they can engage a bigger group, Europe and the US, etc., and try to test that drug. So yeah, there will be drugs out there and you'll be hearing, and we want to encourage you all to just get into a trial. I don't care what trial you get into, but all the trials will give us knowledge in this disease.
Off the topic of atorvastatin, but an important question right now is, have we learned anything from the COVID registry yet? Do we know, especially about neurological issues?
Yes. So the COVID registry has not had a huge number of patients. What are the last numbers?
I want to say it's maybe 12. I haven't looked lately.
Yeah, 12 or 13.
Yeah.
So these are proven cases that have had both proven COVID and proven cavernous angioma. And there has been no case where COVID worsened the known CCM disease, meaning you knew you had the CCM and you had COVID, the CCM never blew up in any of those cases. And there were no thrombosis or neurologic manifestations in those cases. There is one case that's kind of weird. It happened on my service. I operated on it. It came in a person who had a cerebral hemorrhage of unknown cause and was found to have had COVID when he was admitted to the hospital. And his bleed was a cavernoma. So the idea here is that was at the height of the crisis in March when everybody at our hospital had COVID. So I don't know if this was a COVID bleed that just happened to ride the coattail of the pandemic, but that patient doesn't meet the criteria that he had a known CCM and it bled on after COVID. So of the others, dozen or so that had known CCM and suffered the disease with known PCR positive tests, none of you so far have had a neurologic complication. I want to make a call that if anyone thinks otherwise and think that they have a different experience or any experience with COVID and CCM, please join the registry. It's anonymous, and you'll be able to be contacted just to give data, and nobody will ever know your name or your disease or anything, and we will be able to tell the rest of the community more.
Great. There's a whole bunch of questions coming in about microbiome. I think just to give them some broad categories, do you feel like there's a difference between natural emulsifiers versus artificial chemicals that emulsify? An example was eggs. Eggs as an emulsifier, is that a problem?
We don't know, but we don't think so. Because I think the eggs are not known to increase gut inflammation. So the only reason we used emulsifier is not because we wanted to pick on emulsifiers. We used emulsifiers because they are a model of creating a gut leak. They are known to do that. So we added them to the diet of the mice, as if you're taking yogurt that looks like plastic and eating it. And those mice bled from their cavernoma. So, but they bled because of the effect of the emulsifier on the gut. They didn't bleed because so we do not know that eating healthy things like eggs or other things that are not known to alter your gut leak should affect cavernoma.
And then another question is about antibiotics. Should people be concerned about taking antibiotics?
Yes. So antibiotics that kill gram-positive bacteria, things sometimes tetracycline, penicillins, things used for acne, and things used sometimes for chronic sore throat. They give gram-positive antibiotics. those should only be taken to kill the illness. So if you have the infectious problem. So if you've got a pimple that needs one of those antibiotics, you take it till you kill the pimple. But you don't do it over time more than that, because it will help select your microbiome to produce gram-negative bacteria that drive cavernous angiomas. Conversely, if you have a urinary tract infection, that's a gram-negative bacteria. Therefore, please treat it. Do not live with a urinary tract infection and take cranberry juice. Just go treat it. Kill the gram-negative bacteria, because bacteremia with gram-negative infection can be made to bleed cavernomas. So I don't want you to be the one to show me that. But I've seen it with prostatitis in men also. So if you are a man has prostatitis, you got to treat it. You cannot live with letting that gram-negative bacteria live in your system, because it can drive cavernomas. So it's not just the gut; it's the gram-negative bacteria signal from the gut.
So would my PCP know whether or not my infection was a gram-negative or gram-positive bacteria?
No. He would know that, but he would not know what effect it has on cavernoma. I can guarantee you that.
I get that, but I mean,
yes.
if I go to my PCP I have this weird infection, are they going to know whether it's penicillin or whether?
Oh, yes. Definitely. In fact, that's the basic of prescribing antibiotics. So not only the PCP, but the nurse practitioner, the PA, everybody would know. So basically, I think the message that you would take from tonight is that if you have an infection, you're being prescribed antibiotic, you ask them, is this going to kill gram-negative bacteria or gram-positive bacteria? If it's going to kill gram-negative bacteria, it's good. If it's going to kill gram-positive bacteria, tell them, how long do I have to take it? Because I don't want to overdo it. It could harm my cavernomas.
All right. And then the question is also, what can you do to help your gut microbiome? I know we've said that probiotics are not a good choice.
Let me tell you why. Because probiotics in the mice can sometimes rage gram-negative bacteria. So some people might think that's great because it's giving you all these coliform bacteria. That may be great for other things. It may take away gas or whatever, but those gram-negative bacteria is what harms cavernomas. So you might be thinking you're doing something awesome for your gut, and you're actually doing the opposite. So what we're trying to do is really at some point get to the issue of we can measure what your microbiome is like and reassure you. I think that's more important than signing up for fecal transplant. That's not going to happen, okay? So nobody's going to go get fecal transplant. So what you're going to basically do is find out, is my microbiome good or bad? If my microbiome is good, that's fine. If my microbiome is unfavorable for cavernous angioma, then we'll test therapies to change it.
Okay. So at this point, you can't say, go eat yogurt.
I can say, if you like yogurt, don't eat the one that looks like plastic.
Okay.
Has the
All right. And just so someone else asked a question about whether there is a list somewhere, preservatives that should be avoided. Yes, on our website. And as part of our cookbook, we have that. And we are in the process of creating a new CCM Healthy cookbook that includes, that does not include preservatives and includes lots of fruits, vegetables, and meats. Please go on our website if you have a recipe you'd like to share, because we really need them. We would love to share them with the rest of the community. We're going to try to do a new cookbook every year, so we have new things.
And take vitamin D.
And take vitamin D. Make sure. Make sure.
That's another factor that's very simple, where if you're vitamin D deficient, your cavernous angioma is going to rage. So.
And then the last thing that we always share with people is if you have a sleep disorder or sleep apnea, try to make sure that you get that
because of the high PCO2, and that's not good, and low oxygen. So all of that is absolutely true.
Yeah. So we're pretty much out of time here. There are a lot of questions left in the queue. I have copied them. I will try to make sure that we can get answers to those folks that have asked questions that we weren't able to get to tonight. I also wanted to let everyone know, at the beginning of the chat, I put a link to the support group that we're hosting for immediately after the webinar, as soon as we shut down. If you want to go back into Zoom and spend some time talking with Lindsay and Darla and other people that are probably here right now. That's a great way to spend your time. And every Thursday night, we have a support group, and once a month, we host a CCM 101 group that just is an introduction for newly diagnosed patients. So we'd love to see you at one of those.
And I think what I heard tonight is that there is interest in these Q&As more regularly, and I'm happy to continue to do this frequently with you, not just for the atorvastatin trial, but also as a connectivity with the community to share what we know.
Excellent, excellent. Thank you so
Thank
much.
you. Thank you very
Agnieszka.
much.
Thank
Thank
you,
you,
good night,
Agnieszka,
everyone.
and thanks, Connie, for leading us through these times.
Thank you.
Good night.
Bye-bye. Good night.
Night.